Without further ado, welcome everyone, and thank you for joining us today in Advanced Microscopy seminar. We're excited to hear about challenges and opportunities in setting up spatial omics platform. It is my real pleasure to introduce our speaker, Dr. Alvaro Crevenna, who is joining us bright and early from Rome. Dr. Crevenna leads the light imaging facility at EMBL Rome and works on spatial and temporal organization within cells and tissues, combining microscopy, biochemistry, and computer modeling. This session will explore EMBL use of microscopy in spatial omics and approaches to developing open source tools for research. Before we begin, I would like to mention that at the end of the presentation, we will have about 15 minutes for questions and discussions, and you can type your questions directly to a box or can raise your hand and we will open your microphone to ask your questions directly. Without further ado, Dr. Crevenna, the floor is yours.
Thank you, Irina, very much. Let me share my screen. And I guess the ultimate Zoom question, do you see my screen?
Yes, we can see your screen. Thank you.
Fantastic. Let me, okay, so I should have the laser pointer. Okay, thank you very much. And good afternoon to all of you. It's early and bright for me, and I hope my brain is functioning this morning. So what I wanted to let you know today is a bit of our story and sort of highlight some of the things that we've been doing and to tell you what I think is really cool if facilities take up the role of sort of setting and implementing some of these methods. All right, so I've told you I'm the head of a facility, right? So we have facility missions like every other core facility in terms of instrumentation, services, training, and technology development. In particular, I will be talking about our services, in particular, sort of integrate services that go for histology, microscopy, and potentially some image analysis. And the setting for that this morning is on omics. So why is it that we want to do omics in general? Well, obviously, we want to study the cellular heterogeneity in complex tissues, and where we actually would like not only to see these cells, but actually understand and have a sort of a molecular profiling and really sort of map all the interactions that they have in the space and sort of within their context, right? So just to have a couple of definitions, right? So we're going to be talking about spatial omics. And so these are methods that provide whole, whether that's whole transcriptome, proteome information while retaining the spatial location of individual cells that already constitute tissues, organs, and therefore animals, right? So, and of these methods, there is a wide selection of sequencing-based methods that I'm not going to be talking about. There's also like methods that use mass spectrometry. I'm also not going to be talking about. So in particular, I will be talking about imaging-based spatial omics methods. So these are obviously methods that use imaging and microscopy as sort of as the major technique for visualize things. So obviously, when do we want to use spatial omics? Well, this is one case will be where we have some sort of cellular homogeneity or cellular heterogeneity. And obviously, these methods are made to study cellular heterogeneity, right? So we want to sort of really profile the tissue in situ and to understand sort of the cellular organization and architecture of the tissue in itself, right? And so these might be for tissue, for organoids, for cancer. Clearly, this is one of the major applications. For embryos as well, we're doing that. So anything really where there is sort of this heterogeneity. So why do we want to look in particular at transcriptomics? So what we want to do is we want to place the state of each single cell in its environmental context at a specific moment in time, right? So with that, we can then understand the tissue heterogeneity in normal and in pathological conditions. We want to obtain a cell's transcriptional state and correlate that with its function, right? And we want to identify cell types and subtypes and place them in sort of the native context, right? Right. So with that, so of possible methods that are out there, we decided to focus on one particular method, and this is what I will be talking to you today, which is in situ sequencing, right? Or particular hybridization in situ sequencing. This is essentially the method that is behind the Xenium commercial platform. Difference is we decided to set this in-house, right? So how does this work? So what you do obviously is you have a tissue section, you're gonna fix, you're gonna permeabilize your tissue section, then you're gonna then add a reverse transcriptase. So you make from the RNA, you go to the cDNA, then you add your padlocks, you ligate the padlocks here, right? So the padlocks is an oligo that comes and binds, and bond binding essentially the arms, so the extremes of so the extremes of the oligo come and will circularize, right? So they will close, and essentially then the ligase means that now it's a continuous one, and then you use a polymerase to go around this little circle, and this is called the rolling circle amplification. So you end up with many copies of this particular circle, which then you can add these sort of bridge probes that will then allow you to visualize this rolling circle product, this sort of amplified little DNA ball. You add that P and then you image, right? And so how does that look? So, if you add a one color for a particular gene, then you can look at gene 1, and then gene 2, and then gene 3, okay? If you want to look at hundreds, if not thousands of genes, then just having a linear encoding is not good. And so what we use instead is color combinatorics, where we design, okay, a sequence of colors that we are going to assign to each particular gene. And then we're going to be reading them in cycles, right? So we're going to do perhaps 3, 4, 5, 6 cycles. And at each cycle, the gene will have a particular color by design, by construction. And so essentially what we're going to be doing is we're going to be doing this type of imaging sequentially, okay? And so at first cycle, we're going to have, so for example, gene X will be green, then blue, then red, and so on and so forth. So then we can come back, okay, after the imaging, we register all these images, we put them together, and then it's okay, a spot that was green, blue, and red, then this actually is gene X, and then we can decode all the spots present in our images. And so this is essentially what we've been setting up. And just to show you at the beginning, what we have also what is called an anchor. So there, essentially each padlock has two sequences, one that is unique to the gene that you want to look at, and one that is common to all the genes. And you can also sort of play with how you assign these sequences. In this particular case, we have only four genes. And what we're visualizing here is the anchor, right? This is common to all of those genes. And then you can see this is on fresh frozen mouse coronal section, and you can look at the anchor and the DAPI, and it looks good. So essentially you start to get all these different spots. And then you can actually cycle them through the different colors if you have multiple genes. In this case, we only have 4. So you have some spots in green, some spots in yellow, some spots in red, and then you can merge them and see all these different spots. So that was good. And so the next thing that we actually were that we did was to try to compare these with knowledge. And so we were extracting those spots obtained, and then we would actually compare that against the Allen Brain Atlas. And so you can actually see the expression of these things in space and whether they match sort of previous knowledge. And then we did this with another set of genes. This is really just try to understand the technique as we sort of progress and get our hands into the method. Right. So once we did that, then we thought, oh, this is really cool. We can actually do the experiment. And now we want to go a bit beyond. And so we started to make panels for projects. So one of our panels, we wanted to characterize some of the molecular constituents in brains. So essentially we have different neuronal populations. And then this was actually linked to a particular project. So these got published a few years ago. So these worked, okay. And then we were looking at another project where they wanted to confirm some of the things that they've seen with single-cell RNA-seq in terms of the levels of expression for particular genes in some tissues, and then we could actually recapitulate some of these things. And here you see in the control that is sort of there is sort of that particular gene all over the place. Sometimes that's pretty much all you want, just to sort of see the pattern of expression, and then you can also quantify somehow how much there is. And in this case, the exposure to this particular set of antibodies lowers the amount of that particular gene. This is actually a very nice story that got published last year, where they actually look at the effect of paternal dysbiosis on the offspring fitness. Beautiful work, by the way. And here already we started to see that there were genes that we couldn't see. And these got even more evident when we had a sort of a second trial project. Here, what we wanted to do is we wanted to map olfactory gene receptors expression across the olfactory epithelium. So the olfactory epithelium is mostly most of this part here of the epithelium is made by neurons. Each neuron will express one and only one of these olfactory gene receptors, and each neuron has the capacity to choose from more than 1,000 genes, but they express a single one. So we wanted to sort of map these things with in situ sequencing. And in particular, because then there is a mutant where this is dysregulated and then some effects are observed. And so we wanted to map all of these across the space. So this was based on cDNA, right? The technique that I explained, where we actually were going from RNA to cDNA. Then we designed a panel of about 300 genes. This back then it took about 2 years from probe design to analyze results. And then we did see some differences between wild type and mutants, but we actually failed to see the expected results because the technique seems to have a sort of a low efficiency for the expression of genes that we had in this particular case. So what we have learned so far is that works, although now we have moved and changed the chemistry, and now we actually use direct RNA detection, which is much higher sensitivity. And then we decided to actually then open this as a service. So on the platform, what we normally do is you're going to provide the tissue. We design probes based on your list of genes that are interested. We perform the experiments. We can decode those images. We can assign the transcript to cells, which is very similar to what commercial machines do. On top of that, so we hope to provide, let's say, sort of some standard analysis based on what's called the spatial data object. So spatial data is a data frame created at EMBL that is getting more and more used. And so these will actually also be developed not by us, but by somebody else into sort of a more standard software. And obviously what we would like to do with these is sort of really help the user go from the probe design all the way through the experiment and sort of get them started with analysis, which is sometimes still quite work kind of to do. So what we generally won't do is we won't do the bioinformatic analysis. Either there are platforms or essentially there are bioinformaticians in the different groups. And I think that sort of falls into that space. So we are, again, right, we're a microscopy facility. So there are things that we can and cannot do. What we have learned so far is that it works well for high and mid expression genes, okay? Not for things that are either too highly expressed or too lowly expressed. And why it doesn't work for too high expression? So in principle, yes, you're gonna see more. But if you saturate with dots your tissue, then you have a problem when you're trying to decode things. So one thing that you can potentially do is you can tune the number of probes per gene. We normally use 5. And this is essentially what Dexinium does, is they tune from 2 to 8 probes per gene, depending on expression, to essentially try to balance the number of spots that you see with the sort of resulting with sort of the underlying expression level. These, I have to say, is used mostly for cell typing, although you count spots, okay? if you then start to comparing things with single cell, there is at some point where things are going to break, and it's not very quantitative. Okay. The cost per experiment is about 150 euros. This is all homemade, and Google told me that that's equivalent to about 265 Australian dollars. So it has that sort of cost-effective component if you do it in the lab, compared to a commercial experiment that costs the order of 12 to 15,000 per run. Okay, so there is roughly 2 orders of magnitude per run, so essentially per reaction. So one single experiment takes between 1 and 2 weeks. Obviously, you can parallelize, you can start you can start many experiments at the same time. And so it would allow you to essentially have some throughput. Let me say, so we have relied a lot on sort of open source things, and many of those things seem available. They're still they still require a lot of work before they can actually be used on a service setting. Okay, and so there has been an incredible amount of work from our specialist, Marina Peralta, who has done an amazing job really to bring up to speed some of those pipelines, probe design, in particular, where some of the stuff was available, but it really needed a lot of work to really work continuously for many different projects in a service setting. All right, so I've told you what a bit of the story in terms of the service. We are also interested to do other things. So I've told you that sort of the missions go from instrumentation to technology development. And what I'm going to be talking now is in technology development, right? So I'm a strong believer that if things work in a facility, they're going to work everywhere. And I'm going to tell you a bit about that. In particular, there is the issue of how we look at these spots, right? And so we started a collaboration with a local company called Crest Optics, where they developed a structural illumination module. And I thought, well, if it's all about looking spots, perhaps that actually can be good to allow us to sort of detect our spots better. And I mean, sure, like it's not a very good surprise that if you have better imaging, then you can detect spots better, sure, right? So, but and why would we have to deal with this, right? So normally the number of genes that you can visualize will depend on not getting to optical crowding. So that what that is essentially is just if you would look at all the different spots in a particular area, right, you have too many in there overlapping, and then they will crowd the space, and then you cannot sort of reliably identify these things, right? So there are some techniques that rely on a single molecule, and then essentially you look at very sparse data. There's some others, like in this case, the hybrid, so the direct RNA detection, and obviously short, like if you're here, you can identify well your spots. Once you get to sort of high dense areas, then this starts to get a bit more complicated. So techniques like Murfish or Segfish plots, they normally rely on sort of super resolution or sparsity of the data to beat that problem, right? Essentially, they have many, many rounds. So in this case, they have the pseudo colors, which are just imaging rounds, and they do 80 imaging rounds to really sort of make it very, very sparse, the code, and so really prevent that there are overlapping spots in space. And so by that, therefore, avoiding dense areas, so optical crowding. But armed with structural illumination, and in this case, a company here was developing that model and said, well, how would these work for in situ sequencing? So then we started to compare, and obviously we have wide field, we can deconvolve our wide field data, which is commonly done, and is actually done by, let's say, platforms like Xenium and the nanostring now, Brooker. They probably do something like this. Okay, I'm not going to say that they do that because I haven't been involved with the development of commercial platforms, but I guess I would say it's a pretty good bet that they do. So they all are wide field microscopes, which then potentially do some the image enhancement through the convolution of some of the processing like that, right? So anyway, so we can compare our wide field, our deconvolution, and essentially then what you would then get as a resulting image with SIM. And I mean, no wonder, yes, you get better, sharper spots. And then we started to also quantify these things, right? And so you can go from wide field to SIM, and not only the objects that you see are sharper, okay, you can compare the things they actually can visualize better. And when you already have sort of higher intensity, you can actually resolve objects. Like you can see sort of the spot here in the middle that is pretty much absent here. So not only you resolve sort of the higher intensity spots much better in terms of this, the localizations and histogram, you also detect sort of lower intensity spots, right? You do actually bring a lot of detections forward. And in this particular case with this objective, we see a factor sort of up to 2 if you compare the localizations between SIM or deconvolution with against wide field. So in pink is a SIM, so you have essentially about 2. The increasing number of localizations compared to what you normally see with wide field. And so deconvolution increases a bit, but not as much as actually using a structural elimination. This is with the 25x. We were also wondering whether it would be similar with a 20x. So a 20x is probably what's behind some commercial platforms. Again, I think the Cosmics, they I think on the paper that they describe the method, they say that they have an objective comparable to a 10x. Although if you check on the website, what's described is they have an objective comparable to a 20x. They're not using commercial objectives, so you have to believe that what they're telling you is actually what's behind the instrument. In either way, so either the Xenium or the Cosmic have something around the 20x air objectives behind, right? So, and now you can start comparing, and also in terms of more dense areas. So if you look at wide field, I mean, you see sort of these clouds of dot start to look very much like constellations. If you use SIM, then you can sort of reliably see some of these spots much better. And I don't know if you, but if my sort of data of expression needs to rely on spot detection, I prefer these data compared to the wide field data. Again, you can compare this. So when you have very sparse data in particular, so there is a bit of an increase in the spot detection, which is essentially what we have seen before. Now you can see, well, and now you can also ask yourself, well, would this happen also when you have denser areas? And the problem is when you have dense areas, if you run, I don't know if ever you tried, but you have a very dense thing, you can run a spot detection algorithm, okay, and it will tell you something. How reliable that is a very different question, right? And so you can see that you massively boost the number of detections if you have structural elimination, especially in those very dense areas. And again, we can map how many spots we get in with the structural elimination or the deconvolution against wide field. And so at lower densities, there is not that much difference, which makes sense, right? We're not bringing it that much up. But in the moment that actually gets dense, we can see that we get up to 4 times more spots with the structural elimination that we get with wide field. So, and then again, right? So just to repeat in this, we also extend it towards a 60x, right? So when you have wide field and it's sort of sparse, then wide field, confocal, deconvolution, essentially, or SIM, they all roughly look at the same thing, right? It doesn't really matter whether then you increase the numerical aperture, you're pretty much seeing the same thing. Maybe there's an extra spot here that looked like a dirt or something around, and then you can sort of start discerning these objects here. There will be sort of a minor increase. The big sort of improvement is then when you have very dense objects, okay? And again, so if you would run a spot detection algorithm on data like that, it will speak something to you, but I guess you shouldn't believe that much. It's only when you then start not only improving the imaging, but also improving the numerical conversion, so the collection efficiency of signal, that you will then start to see the underlying spots. So the 60x, I think, is only used on the MERS scope, and so that would be comparable to what you would get. And if you actually go then towards structural elimination, then you see you have a much better resolution of all these different objects in there. This works actually because you have enough signal with in situ sequencing. You have this sort of enzymatic amplification, which you won't have if you're using sort of single molecule techniques. All right, so I've told you that sort of structural elimination improves single transcript identification, then you can do that with the different objectives. But essentially, if you have very dense areas, you are going to need sort of better imaging, both in terms of the objective that you use and the modality. And so one of the advantages of doing this within a facility setting is that you can tune the imaging depending on the needs of the sample. Right? Not every sample would be fine with a 20x. Not every sample would need the 60x, and we can choose how do we image the different samples depending on the panel size, depending on the tissue type. If it's very dense, then we can probably go towards the 60x. If it's sparse and we have sort of cells here and there, so normally brain is good enough to look at with 20 or with 25x, but we can tune those things, which is actually not possible on a commercial platforms. All right, so there was one story that that we worked. This actually was published a couple of years ago. There is a new collaboration that we have in-house, where we actually trying to correlate X-ray tomography to targeted transcriptomics. This is a collaboration with two groups, the group of Elizabeth Tuck, that they're doing X-ray high throughput tomography in Hamburg, and the group of Christian Tischer, who is the sort of the head of the Center for Bioimage Analysis in Heidelberg. So what we're aiming to do here, right, is we want to use X-ray nanoholotomography, which is a non-destructive whole biopsy sort of sample imaging technique that will obtain, let's say, morphological information, and then we want to correlate that with spatial transcriptomics, right? So this actually would be very similar to, let's say, doing H&E in spatial transcriptomics, which is very, very possible to do. But now what we want is we want sort of a whole sample kind of thing. So to actually look at it in 3D. So why do we want to do X-ray tomography? Well, we can look at 3D whole biopsy samples. It's non-destructive, it's fast, and it's fast also because it has been fully automated. We use FFPE tissue, which essentially means that it's compatible with clinics and biobanks. X-ray tomography has isotropic resolution in contrast to light microscopy. And in this particular case, depending on the brightness of the beam, you can achieve cellular resolution. How tomography works. So essentially, you're going to put the sample here on a pin. It will be a little thing there hanging. And then the beam comes out of this side on the diffractometer, essentially hits the sample, and then it will you will hit the detector. And what you do is for each position of the detector, you take a full rotation of the sample. So this rotates, and all these images are being acquired. And then you move the detector to a second position, you rotate and acquire all those images, and you do that essentially 4 different times. In this particular case, we're using the P14 beamline. So this is about normally 12.5 roughly kiloelectron volts. This is done at the synchrotron in Hamburg. And so what can we do with that is that you can reconstruct 3-dimensional volumes, okay? And so this should work, maybe. Yay. So this is I know it stops sometimes. So this is a brain biopsy. This is a brain sample. NFFP is a block, and then essentially they punched it, and then we can actually go through. And I don't know why it always stops. There you go. Okay, and so you can actually go through the whole volume, and you get morphological information. And so then what we want to do is we want to retrieve those samples back, and then perform spatial transcriptomics, and this seems to be working. And so we can then do multiple cycles of imaging with different genes. So we have a neural panel, and then we can sort of detect all the different transcripts. We can overlay these images, and ideally, then we want to bring this molecular profiling back into the morphological data. And then one can start thinking whether if you know some of that for some slices, then you can sort of extend, use some fancy AI to sort of decode the entire cellular heterogeneity in the tissue. We can also develop 3D protocols to work on entire tissue samples, and you can really try to merge the information that you get from morphology through the X-ray and the molecular profiling, right? And so because this is not limited to transcriptomics, then you can also add, obviously, antibody-based markers. So this has just started. So obviously, we want to optimize these protocols for FP. We want to develop and automate some of the experimental pipelines. We want to develop all the computer analysis needed for registration and for the analysis of both morphology and the molecular profiling. We want, this is something that has just started, is we actually want to develop experimental products that will allow us to work with the whole biopsy so that you don't need to section. And these mostly will probably work just for cell typing. And as part of this project, we will design whole transcriptome, both human and mouse probes. So, and then make them available. So if anyone else wants to then look at particular genes, say the sequence of those probes will be available. All right. I think I'm early and I'm going a bit fast. So the last thing that I want to tell you is, so these projects have been looking at, let's say, technology development, right, internally. And so the last thing that I want to tell you is how we are engaging with external partners. So I've mentioned the fact that that we had a collaboration with Optics, where we then that allows to go from sort of these blurry things to sort of more accurate detection using structural elimination. One thing that we would also like to improve is on the 3D imaging, especially because, I mean, this is sort of bread and butter for place for where you look at, tissue and embryos and all these things. Everyone knows that as you go deeper, you lose signal, and even you have beautiful images in X and Y. When you make the volume, these look ugly, right? So what we need is adaptive optics. So we're working on that. I've told you that we are combining RNA detection with our X-ray imaging. We're also combining RNA detection with chromatin tracing to be able to relate sort of chromatin conformation techniques with RNA. This seems to work. We're slowly making progress. We also develop a spectral mixing algorithm to increase the amount of colors that we have for combinatorics, right? So the combinatorics in spatial transcriptomics depends on the number of colors that you have. If you then increase the number of colors, then you will be able to sort of more genes. So similar to the adaptive optics, but then potentially using deep learning approaches is just to do sort of reconstructions where then you can try to recover the isotropy of the volumes. This is normally called digital adaptive optics, as well as we then have a custom fluidic robot for all these exchange experiments. Right. So I've shown you how a facility can collaborate with a company to develop technology and sort of highlight its power with the methods in-house. I believe that this could be a general thing, okay? And in particular, I believe that core facilities can be innovation partners. And so I wanted to create a space dedicated to co-create technology with industry partners. So what we're doing at the moment is, so I've told you the Optics in particular. So they took this when it was an alpha system back then. Obviously, I couldn't talk about that, but now it has been released, so now I can. And so this model is cost effective compared to a full system. It's an add-on, right? there we go. It's sort of light efficient, so you can use sort of lower exposure times. And I have to say it was not as optimized as the size, although it can actually work with lower magnification objectives. I think the size now can do that. I'm not sure. At that moment, the one I had wasn't able to do any of that stuff. And essentially what it happened is that, as I said, so I had a size system there as a demo, and I was asked to test in that moment, alpha version deep sim model. And I gave a lot of feedback, and I was very happy to see that some of that feedback got integrated into the final commercial product. And it's just a comparison we can see, and then let me just make this bigger. So the confocal with the 20x, right? So this is a confocal with a 60x, and you can actually use you can look at the SIM module with the 20x. So obviously this is a zoom, so that you have the same field of view. The nice thing of using the 20x in tissue is that you preserve sort of the large fields of view, and so. So for sort of large sample imaging, this makes it very attractive. The fact that you can combine both the sort of resolution with the large fields of view. So seeing that this was actually quite positive, we decided to engage with other companies, and so we started collaborating with a very small startup. So I guess Crest Optics is more of a medium-sized company. Flim Labs was at that time, I think there were two. It was Alessandro Rossetto, the CEO and founder, and maybe there was somebody else on the company. And so they had this little box, which I have to say was not the prettiest thing. That box has now evolved into a sort of a nicer looking electronic box, and now there is a full upgrade kit, and this allows that where you can then have a sort of a full upgrade of your scanning confocal into FLIM, and so you can add the lasers, the detectors, and all synchronized with electronics, and it's quite competitive in terms of pricing. And you can export. There's an API and all these things, and you can try them. So this has also been quite successful. So all in all, made me think, oh well, maybe we should have dedicated space to do this type of activities. So at the moment, we are creating an imaging innovation lab where we want to focus obviously on the collaboration and the technology co-creation and facilitate adoption, right? So normally we're taught in sort of innovation courses that there is this sort of value of that where you have resources in academia, and then obviously once you have a big established company, then they can access funding to develop new things. But in essence, there is sort of this in between where there are very little resources, and where I wanted to place myself as let's say as a facility or as a partner for these companies is really to try to help them facilitate adoption, test with real sort of real world cases, and we could co-develop things together, right? So obviously the focus will be things that capitalize on local research and promote open source tools. There's a list of potential partners, and if you haven't heard from them, I think it's good because they are small, and the whole thing should be funded by industry partners, shared with CNR, which is the local Italian national research system, and these will all be managed by EMBL. So these should be coming up soon. And with that, I really want then to thank the current team, Emerald, Marina, who has done an amazing job over the past year to really bring a lot of the methods and techniques and pipelines sort of from an academic level to sort of a service ready level. Daniele, Gerardo, Julian, Erika, and past members Ines, Elena, Sergio, and Alejandro. I have to say, all of these would not have been possible without the collaboration from Mats Nilsson and Marco in SciLab Lab, and help from Carolina to also set up some of the analysis pipelines. The X-ray tomography to transcriptomics is a project shared with Christian Tischer and Liz Duke, thanks to funding from CCI. Just as a commercial, so we run actually a practical course every 2 years. So this started in 2023. Then we had an edition this past summer in Heidelberg, and we're thinking potentially organize one here in Rome in 2027, or potentially take it to somewhere else. And the last thing is, if any of this is appealing to you, there are several training opportunities at EMBL. So there's the iPod Link, which is a sort of the postdoc program that is actually shared with a partner. There's the RISE. So we actually have a dedicated postdoctoral program in technology development. And we also have visitor programs. So if you want to come for a sabbatical, we have technology development internships, and there are a few other fellowship schemes that allow you to either visit labs or facilities if anything that is done across EMBL is interesting to you. Do not hesitate to reach out. And with that, I take any questions that you have.
Thank you very much. That was very exciting to hear all the lovely developments that's happening, because obviously, as we observed that space, it was obvious that microscopy setup would be best suited to pursue the space, that space, and the tools that you outlined here highlight how much better result we can have when apply those tools. I cannot see any question on the question line. Would anyone like to ask? Want to ask a question? Please raise your hand, and we will open your mic. Yes, I can't. I can't see that. Alex, can you see?
No questions are coming
No.
up, but is it maybe if do you have any questions out there? You can answer. You can just type in the Q&A. Doesn't look like anyone has any questions. Alvaro, how easy was it for you to communicate with the companies? Were they quite, easy to communicate with? Was it an easy partnership for you during this time?
I mean, I guess you have different levels of commitment. I guess it's a bit based on I took a social psychology course not long ago, and there is a technique that is called the foot in the door. And so you start with something small, and then you can build up, right? So one of the things that company, I guess, and I understand that they're very wary, is where they put resources. So compared to maybe to academic labs where you're always playing with different things, commercial entities have to sort of focus what they do, and certain sort of research and development activities are a bit riskier. But if you then prove that you're sort of a reliable partner, then they're much more willing to engage with you. So you always have to find sort of a good project in terms of that something that it interests them, but it's riskier, or it's risky, so that you can de-risk the project. Essentially, your participation will make that actually you lower the risk, and then they will actually be very happy to engage in collaborations with you.
Okay. Did you have to suggest why you thought that was like a niche area that researchers would actually respond to? Because I guess my question would be like, is the company coming up with are they seeing the gaps that they're filling, or did you suggest the gaps that their technology could fill in the market? Like, as the expert in sort of microscopy, are they relying on you to say this is where you should go, or did they actually suggest it and you come up with the samples and the experiments to test the product?
So I guess it can happen both. So I can tell you in terms of the structural elimination module, I had the competitor one literally next to it. And so for my applications, I knew what was missing in that other instrument. And so I suggested a bunch of features that I wanted. I guess in a very, self-centered interest kind of way, but I say, look, I don't want that. I want this, this, and this. And they took some of those. I mean, at the end, I don't think that they completely only rely on me, but I want to believe that I had an influence into what they actually did.
Yeah, no, I think it's a good partnership for sure. I do see a question from
Yes.
Tess.
I can read the question.
We can allow it to talk. I don't know, Tess, do you want to ask a question directly?
Hi guys, thank you. My question was, what was the rationale for building and developing in-house spatial rather than adopting a commercially available platform, which seemed to be everywhere at the moment?
So the short answer to that is there wasn't when we started. So we got interested in spatial omics, spatial transcriptomics in particular, about 6 years ago. They had just been published the paper on Segfish Plus, but there was no commercial platform. And actually back then there was also another company, was called Rebus Bioscience, that essentially went bankrupt. So at that time there was no commercial platform, and we started to do things. I think down the line, honestly, we will probably have one or another commercial platform. At this moment, I guess we still like the flexibility, the degree of customization that you can do, the fact that you can choose how you image your samples. That is just so much better for an academic setting compared to just relying on those things. We also have looked at some data from companies and from the ones that we can get images out, and we have our concerns of how well they're actually decoding things. So I believe, honestly, that things will get much better also in the commercial setting, and hopefully also prices per run will go down. And so one aspect, for example, that I've tried to do in the facility is we are not trying to cover, let's say, whole transcriptome. So commercial platforms, I go in sort of that direction, right? I have the front, what is it? This the 5K prime, the whole transcriptome from nanostring, now Bruker. That's something we don't want to do, right? I don't want to sort of scale up in the number of genes. Why? Because I see those experiments as exploratory, okay? And normally once you've done that, you have narrowed down your number of genes to something on the order of a few hundred, maybe. But that's the least that you then want to look over and over and over, and many experimental conditions, mutants, whatever, replicates. And if you have to run all of those different experiments on commercial platforms, that's just going to be a massive loads of money, which you may have, right? You are very welcome to go and do them. But if not, right, if we can take some of that, obviously, it allows you to scale and throughput more than scale the number of genes that you can see. And the other thing that we've seen is that then it is essentially having the capacity, this is from a sort of facility point of view, having the capacity to run these experiments, it also allows you then to say, okay, then we can combine this with protein. We can combine these with chromatin tracing. We can combine these with potentially you can combine two different spatial transcriptomics techniques. That's also something that we're playing with. So it builds capacity internally, which down the line for research groups, it will play a role and it will benefit them.
I don't see any other questions. Ishka, do you want to?
No, I just wanted to add that obviously from various symposia, what we can see that the researchers are looking exactly for that type of flexibility. It's much harder to deliver such flexibility on the commercial platforms. And also from our perspective, we understand how hard it is to perform those experiments from a costing point of view once you narrow your targets and to have opportunity to deliver wonderful science with much less cost. I could only see that will be very desirable for researchers. Yes, and I would like to thank you all for joining us in that seminar, and in particular our guest speaker who had to get up very early to deliver that most interesting seminar to us. And thank you very much for joining us. Thank you.
Yeah. Also, thanks from my side. And don't hesitate to reach out with any questions you may have.
I would like to also add that seminar will be we record and is available to people review later, or for those who could not join us today, they will be able to watch that seminar later on. We have a YouTube channels MMI Advanced Microscopy Seminar YouTube's channels when you can watch those seminars. Thank you yet again. Thank you, Alvaro.
Thank you.
And good afternoon to you all.